Right, good evening, everyone. This evening we have Dr. Nessa Carey, who has spoken to us before. She has her own Wikipedia page, if anyone's interested. In fact, she's got many pages on Wikipedia, so it's probably worth a look. Post talk this evening. Dr. Carey is a British biologist working in the fields of molecular biology and biotechnology. Expertise in the field of epigenetics and in technology transfer. And I think it's fair to say that this is practical application of academic study, so that transfer between academia and industry, I guess we'll touch on this evening.
Let's just get going.
You could do. Yeah, I don't know whether we're talking about destiny in terms of the metaphysical or the microbiological, but I'll leave that to you to decide this evening, and I will hand over. I think Barbara, when she wraps up, she'll be able to talk to the next lecture. I need to dash again this month, so apologies for that. But enjoy the talk.
Thank you very much. I'd just like to point out I didn't have a sudden rush of ego to the head and write myself a long Wikipedia article. Somebody else wrote it. And I think it's part of the campaign to get more women scientists represented on Wikipedia, which is very nice of them. Unfortunately, almost all of it's wrong. But apart from that, it's absolutely grand. So it's lovely to be here with you this evening. And I get the chance to talk about epigenetics again. I love epigenetics. It is the weirdest, strangest part of biology, and yet makes perfect sense and answers loads of questions that we never really understood and never even asked very well. And so I'm going to take you through it this evening. Talking about epigenetics, which, as I'm sure you've worked out, probably has something to do with genetics, with DNA. And for quite some time, we were sold the idea that DNA codes for who we are, that it's the explanation for biology. And I wouldn't want for a moment to suggest that DNA is not important. DNA is incredibly important. If you look at that little girl who's dressed as Snow White, she is about 7 or 8 years old. She already has the wrinkling skin and the hair loss of somebody in their 70s or 80s. Her cardiovascular system is starting to develop large amounts of atherosclerosis. She will die before her early teens because she has a condition called progeria, which has a very severe form of accelerated aging. And it's extraordinary to think that that is caused by just one wrong letter in the DNA alphabet. Like the rest of us, she inherited 3,000 million letters of DNA from her mother and 3,000 million from her father. And one of them is wrong. 1 in 3 billion is wrong, and she gets this devastating disease. So sometimes genetics can have an enormous impact on us. And it doesn't have to be extreme as that. If you look at the coloured blobs, there's about 30 men in the audience, so there's about 3 of you going, why are there coloured blobs on average? Because about 3 of you are probably red-green colourblind, and you can't see that actually up there is the number 16. And that's genetic as well. It's 1 incorrect letter in your genetic code.
16 is 26.
No, it's 16. Definitely 16. Yeah, that's not red, green, colourblind. That's just yeah. Arms too short, arms too long. So again, that's a genetic effect. So genetics does influence us. I'm not for a moment becoming this epigenetic zealot who says it has no impact. Genetics absolutely does influence us, but it can't be the only thing. And for a really long time, we knew that genetics couldn't be the only explanation to the diversity of human life. We just pretended it didn't really matter, that it couldn't really be the answer. We just carried on as if it was. But we've known for a really long time, long before we knew the human genetic sequence, that actually it couldn't be. If you look at schizophrenia, right, devastating disease. It affects about 1 in 100 of the population, a remarkably consistent level throughout various populations, 1 in 100. But if you have a relative with schizophrenia, your risk of schizophrenia is higher than 1 in 100. And the more closely related you are to someone with schizophrenia, the higher your risk becomes. if you have a non-identical twin and they have schizophrenia, your risk jumps from 1 in 100 to about 1 in 6, it's 17%. If you have an identical twin with schizophrenia, your risk goes up to 50%. Your risk is 50 times higher than the general population. That's a terrible way of doing it statistically, and I apologize to Pauline about that, but basically your risk of schizophrenia is massively higher. And that tells us that genetics definitely plays a role in schizophrenia. But the interesting thing to me is it's not 100%. And so it tells us that even two genetically identical people, two people with exactly the same DNA alphabet, don't have the same risk of schizophrenia. Something else must be happening. And wherever you look in biology, you get these indicators that something else must be happening. So if I were to take DNA from this gentleman, DNA from this lady, and put them through a DNA sequencer, I could work out which DNA came from the gentleman and which from the lady. Never, by the way, do this in a dark lecture theatre, because you can get it wrong, and it's really embarrassing. But you would be able to tell, because in humans and all other mammals, males have an X chromosome and a Y chromosome, and females have 2 X chromosomes. If I take DNA, I'm not really going to take your DNA, it's all right. Yeah, I know. If I take DNA from a male turtle and a female turtle, I cannot tell genetically by sequencing the DNA which came from the male turtle and which came from the female turtle. Because in turtles, sex is not determined genetically. It's determined by the temperature at which the eggs incubate. One of the many devastating consequences of global warming is that after those turtles as a species surviving for hundreds of millions of years, we may be wiping them out, because there are now parts of the world where 95% of the turtles that hatch are female because the sand has got warmer. So in turtles, something as fundamental as sex is not determined by genetics. If you look at honeybees, queen honeybees and worker bees are very, very different. Not as different as you might think. I have bees and I can never find the queen, though I know she's in there somewhere. But they are actually very different, and queen bees will live 20 times as long as worker bees. But again, if you take the DNA, there's nothing at the DNA level that actually distinguishes queens from workers. It's all to do with how they're fed at a particular stage in their lifestyle. So the idea of genetic determinism is actually on pretty shaky foundations. We have hundreds of examples like this. So there's got to be something else as well as DNA to explain the diversity of what happens in biology. And one of the best examples of how DNA cannot possibly be the only explanation is every single one of us in this room. We contain around 70 trillion cells each. And with the exception of a tiny percentage of cells in the immune system, those 70 trillion cells have exactly the same DNA sequence as each other. Absolutely identical. And yet the cells are phenomenally different. So that neurons and fibroblasts and gut cells and other cells look completely different and behave completely differently. And they always stay as those sorts of cells. They don't turn into each other. And yet they have exactly the same DNA code. So there must be other things that influence us as well as our DNA code. And until about 30 years ago, if you said to a scientist, well, why? Why is it that we have all this DNA and so on, and it's not leading to identical outcomes? A scientist would give you some version of it's nature versus nurture, that it's the combination of genes and the environment, that even the environment your cells develop in the embryo varies, and therefore cells vary, which is absolutely true and sounds wonderfully scientific, and is actually just a more complicated way of rephrasing the question back at us. It doesn't tell us anything, it doesn't explain anything, it's just a description telling us what we already knew. Why epigenetics is so much more exciting now than it was 30 years ago is because epigenetics actually gives us molecular explanations for how this happens. How can you take the same DNA sequence and end up with completely different outcomes? Epigenetics provides the explanation for this. And these are various representations of epigenetics. That's on the right-hand side. And I could go through it in enormous detail, but I've never found any of these models particularly helpful. And so I created my own. I apologize to anyone who's seen it before, but I have never found a better way of describing this, because I made my model out of sweets. I found it by far the easiest way of creating a model of the molecules of epigenetics and describing it to people. It also had the advantage I ate it once I'd finished, which I really, really liked. So imagine DNA, our fantastic genetic material, our 4 wonderful bases that we get 3,000 million of from each parent. Imagine that is the strawberry laces, okay? Nice, long, stringy molecule. Now, this nice, long, stringy molecule, whenever you see DNA pictured, it's always, will it go backwards for me? Yes. Let's do the previous one. It's always pictured a bit like this one on the left. It's always pictured as this long, double-stranded, stringy molecule that goes on and on and on and on and on, like a really curvy railway track. Trouble is, that can't be how it is in our cells, because that long, stringy molecule would take up about 2 meters. Very, very thin 2 meters, but 2 meters. And the nucleus of our cells that contains DNA is only about 1,000th of a millimeter in diameter. So it can't be this long, stringy molecule. It must be very structured and compacted and kept nice and tidily in the cells. And the way that this works is that DNA is wrapped around proteins, cunningly represented by the marshmallows, right? So what you get is you get 8 marshmallows, 8 proteins. They're called histone proteins. You get 8 of these in a cluster, okay? And DNA wraps around them. So DNA wraps around 8 of those proteins, and it carries on a little bit, and then it wraps around another 8, and another 8, and another 8, and you get this lovely structured organization. And there are millions of those clusters of 8 histone proteins. So those proteins provide a way of structuring DNA, but they do so much more. They also control DNA. They control how it's expressed. Now, I can't help noticing that quite a few of the people in the audience are more at my end of lifespan than perhaps the people in the back row, right? So I'm guessing quite a few of you have grandchildren. And when your grandchildren were little, you did that thing of going, of course we'll take care of the little darlings over half term, right? And the little darlings turn up and it's great for the first day. And by about Wednesday of half term, you're thinking, dear God, when does it end? And you think, I need a little gin. I need to take the edge off. I've got the little darlings to bed. I'm going to have a little gin, take the edge off. Let's say it's even worse. Let's say it's not half term, it's summer holidays, right? Yeah. Right. So your little darlings are with you and you're getting really quite tired as the summer holidays go on. And so you think, I need another gin, take the edge off. Only by the time your grandchildren have been with you for about 10 days, it takes much more than 1 gin to take the edge off. It takes several gins to take the edge off. And this is because your body has adapted. Your body has adapted to the fact alcohol is coming in, and it has started expressing the gene that codes for the protein that breaks down alcohol. So this, I once went on holiday with surgeons. My alcohol tolerance by the end of 2 weeks was extraordinary. My God, not even drinking. And the reason is taking more alcohol, signals go to your liver saying, there's an awful lot of this poison coming in. Could we create detoxifying enzymes faster? And this is what happens. This is how it happens. Those signals get to the liver. And there are lots of steps in this process we don't understand. But what we know is they get to the liver and they get somehow, and this bit is really vague, we don't know how this happens at all, they somehow affect the histones, those 8 marshmallows, that are next to the gene that codes for the protein that breaks down alcohol. So we've got that gene, that one particular gene, wrapped around those marshmallows, and a signaling cascade is set up, and tiny modifications get added to those histone proteins, represented by the green jelly dots, right? And what those modifications do is basically signal to the cell, we need to switch on this gene more highly. We need to produce more of the protein to break down alcohol because there's a lot of alcohol coming into the system. So that gives you a way of increasing the amounts of this protein. Now, let's say it mercifully gets to the end of half term or summer holidays, and the grandchildren, who no doubt you will miss terribly, go back to their parents, right? And after a little while, you think, I should probably cut down on the gin now. I've heard it's not very good for me. And so you cut down on the gin. Now, at that point, there is no point the liver producing large amounts of the protein that breaks down alcohol, because there's no alcohol coming in, it's just a waste of resources. Signaling cascades are set up again. And those green jelly tots that said, those green modifications that said, turn the gene on, are replaced by different modifications, represented by the purple jelly, my personal favorites of all the confectionery on these slides. And those signal to the cell, start turning this gene off. We really don't need such high levels of expression of this protein. I've used two different colors of jelly tots here. In reality, in the cells, there are 60, 70 different types of modifications that can be put on these histone proteins, and they can be put on in all sorts of combinations to have very subtle effects on the expression of that gene. But sometimes if you have a lot of the purple ones, the ones that say, let's turn the gene off, what it can also do is it can attract even more modifications, but this time to the DNA itself. And these modifications get added to the DNA, and that's saying, no, seriously, we don't need to express this gene. So let's turn it off. There are some regions of the genome, of our DNA, where you get unbelievably heavy levels of this modification, the orange one. And it actually distorts the whole shape of the DNA. And the DNA gets completely squashed and compacted, and that gene just cannot be switched on. And so we have a way of controlling gene expression. Our cells have a way of controlling gene expression. And there are certain things that it's really important to understand about these modifications. They're tiny, they're really small chemical groups, and they get added either to the DNA or to the proteins, and both. They never change what the gene codes for. If the gene is coding for the protein to break down alcohol, the gene just keeps on coding for that in exactly the same way. It doesn't change the sequence at all. It doesn't change those letters of DNA. It changes the levels of expression, and it can be passed on when a cell divides. So the same pattern of DNA modifications get passed on, but often the same pattern of modifications to the histone get passed on to the daughter cells. So what this means is that the epigenetic system, because this is what these are called epigenetic modifications, they can act as an on-off switch. They can turn a gene on, or they can turn it off. They can silence it. And they can silence it in all the cells that that cell divides into. So this is why the cells in your liver stay like liver cells every time they divide. This is why the cells in your skin, when they divide, create more skin cells. They don't start creating kidney cells. This is why you don't get teeth in your eyeballs. I wanted to call my first book, which ended up being called The Epigenetic Revolution. My working title for it was No Teeth in Your Eyeballs. And my publisher said, and I quote, Nessa, it lacks gravitas. I still think it would have sold much better. Anyway, so you can turn off genes for ages, forever, and that controls cell identity. Or you can have regions, particularly where there's the histone modifications on the marshmallows, but not the ones on DNA, where the gene can be switched on, but the body uses epigenetic modifications to control, is it expressed a lot or is it very little? It's like a volume switch. It gives us enormous flexibility in how genes are expressed. So we shouldn't really just think about the genes and the environment. We should think about how do genes and the environment talk to each other, and they do it through epigenetics. They do it through these modifications. So we have this circle of the environment changes the epigenetics, and the epigenetics changes gene expression. But the gene expression can then change how we respond to the environment, which in turn can reinforce or alter the epigenetics. So we have these cycles of activity. And we also have to factor in as well that sometimes we also get mutations in our DNA. We get very rare changes in our DNA caused by things like chemicals or ultraviolet light. So how we end up is a combination of all of these factors. It's a combination of what genetic sequence we had to begin with, how that got affected by epigenetics, how the epigenetics was affected by the environment, and also did we get some random mutations. So it's an amazingly complicated, beautifully elegant system that explains stuff that we simply couldn't explain 30 years ago, which I absolutely love. But what I also love about it is we're starting to do practical things with our understanding of epigenetics. This drug here, that interferes with the modifications that are put on the marshmallows, on the histone proteins. It actually means that you get more, let me get this right around, histone deacetylase in, so you don't, yeah, you get more of the green jelly tots, right? You get switching on, you get certain genes switched on. And this drug, which we now refer to as an epigenetic drug, is now used really successfully in the treatment of people who have certain types of blood cancers, called peripheral T-cell lymphomas and cutaneous T-cell lymphomas. It's really, really good at controlling those cancers. And one of the reasons I use this example is that I played a role in the development of that. I know. Okay, thank you. Actually, my role was tiny because, you know, it costs hundreds of millions to develop new drugs and it takes massive numbers of teams. But it is still actually the thing that I like most about my career, is that somewhere along the line, I have made a difference, which is one of the great things about working in science. So we can do great things in terms of creating drugs by using our knowledge of epigenetics. We can also understand disease better. One of the things that the UK has always been brilliant at is setting up cohort studies where we follow people for decades through their lives. And one of the things that became apparent through these cohort studies is that often disease starts very early. It's something that's called the Barker hypothesis, because it was Dr. Barker who identified it. And it's called the fetal origins of adult disease. That we know that actually stuff that happens in utero when a baby is developing can have a really major impact on their health for the rest of their lives. So if the mother is very overweight, that can have an influence on the baby's future health. If they drink alcohol, if they smoke when the baby's gestating. And we really couldn't understand how this would happen. We knew it happened, but we had no insights into the mechanisms of it. Epigenetics gives us a really good set of theories that we can now start investigating. Because the theories now are that in those early events when the baby is developing, they change the epigenetics in certain tissue systems, and that then influences how that baby's cells will respond to life events as the child gets older. Because the epigenetic modifications that get established when you're very young actually can be very long lasting and can influence you for years. I'm not saying epigenetics is the only answer to this, but it is almost certainly part of the reason why fetal health is so important for later life. Some of you may have seen the reports recently that sugar in the first 1,000 days is linked to poor health later. So that 1,000 days includes in utero. So sugar in the first 2.5 years, which makes me feel slightly bad about all the jelly tots and the marshmallows and stuff. But on the other hand, I never share sweets with children anyway, so yeah, it's not mine. But yeah, until we started to understand epigenetics, we had no clue how it could possibly be that events that took place before you're even born can be affecting you 50 years later. It is almost certainly involving the development of epigenetic markers that are influencing gene expression in ways that turn out not to be helpful. So it allows us to have new ways of approaching health problems and health questions, which is fantastic. But I also think I did mention that one of the things that I really like about epigenetics is it's just plain weird, right? And I love that. I love the weirdness of biology. So these animals, absolutely love these animals. Stick insect, a nurse shark, I think, could be a leopard shark, can't remember, nurse shark, leopard shark. A beautiful salamander, my absolute favourite, the Komodo dragon. Love Komodo dragons. Wouldn't necessarily want to meet one down the dark alley, but yeah, absolutely love them. And a Californian condor. all of these species, plus many of their relatives, are able to do something that no mammal has ever done. Anyone know what it is? Some of them can, not all of them. But the thing where animals change sex, that's absolutely epigenetics. Anything else?
Change colour.
No, some of them can. That doesn't tend to be epigenetics, but nice try.
Regrow a limb.
Some can. Yeah. Okay, if I was going to say the word Venus, not the planet, the goddess. Not helping at all, is it, Athena? Okay, let's say Athena. And I'll tell you what. They have all produced young without ever having mated. Yeah, I know. I know. In fact, if someone says to you, would your grandchild like a stick insect? Say no. Because you will end up looking after hundreds of stick insects because they will lay fertile eggs. All of these species have given birth to live young without having mated. Mammals don't. A mammal has never naturally done this. And for the longest time, again, if you said to scientists, why don't mammals ever create live young, have viable offspring without having mated? They would say things like, well, there's a barrier. There's a barrier that prevents parthenogenesis. It's like, yes, I know that. That's literally the question I just asked you. Why does it happen? For ages, we only had that description. Now we have the explanation, and the explanation is so beautiful. So beautiful. And this is work done by a fantastic scientist in Cambridge called Azim Surani. I had the privilege of working with Azim, and he is just extraordinary. And he has won everything except the Nobel, and nobody can understand why he hasn't won the Nobel. So if anyone ever asks you, if we ever turn the Nobel into like Strictly Come Dancing, where you can have audience vote, can you please vote for Azim? Because he really ought to get it. Anyway, Azim did these beautiful experiments. He had actually been trained by Bob Edwards, who is the guy who, there was the recent drama about in vitro fertilization and the first test tube baby, Louise Brown. Well, Bob Edwards was the man who wrote about the technology. So Azim Surani had been trained by him, so he used in vitro baby technology. So what he did was he took a fertilized mouse egg, right, and he took the nucleus out. So there was no genetic material, there was no nuclear genetic material in them. And then he injected into that egg 2 egg nuclei, and they fused, and the embryo began to develop, and he implanted it back into the uterus of the mouse. And development went on for a bit, and then it went absolutely haywire. No offspring. And then he did the same experiment, but instead of injecting 2 egg nuclei, he injected 2 sperm nuclei. Same thing happened. Development started, then went haywire, actually even faster. And then did the same experiment, which is essentially the classic in vitro fertilization experiment, where he injected in an egg nucleus and a sperm nucleus. Yay! Live offspring once this has been implanted back into a female mouse. That seems very obvious. That's like, well, that's just a more technical way of telling us. It's exactly what you'd expect. Yeah, Azim did something so beautiful. In each of these experiments, he set them up because you could use genetically identical mice to get the eggs and the sperm from. He set them up in such a way that if you had the experiment with the 2 eggs, or the 2 sperm nuclei, or the egg and the sperm nuclei, those 3 conditions were genetically exactly the same. There was no difference at the DNA level. What Azim's work showed is that mammalian reproduction is critically dependent on something which is not DNA sequence, because in those 3 experiments, the DNA sequences were identical. He showed that the barrier to parthenogenesis in mammals is not to do with DNA. It's to do with some other bit of information. And the other bit of information, as he speculated and showed in his work, was epigenetic information. It's about, you remember the orange jelly tots on the DNA? Well, some bits of mammalian DNA in egg and sperm come pre-marked with those orange jelly tots. They come pre-marked with epigenetic modifications that say, I'm from mum or I'm from dad. And those have to be right. if you haven't got the I'm from mum and I'm from dad, you can't get the offspring. No mammal would exist were it not for epigenetics. And sometimes this process can go wrong. So we see children who are born with quite significant, particularly intellectual disabilities. The little girl here has something called Angelman syndrome, and the boy has something called Prader-Willi syndrome. And in both cases, there are problems with appetite, there are problems with being able to gain weight or being unable not to gain weight, and there are problems with educational disability. Their DNA is absolutely perfect. There's nothing wrong with their genetics. It's their epigenetics went wrong when the egg and the sperm were being created. So epigenetics absolutely has a major impact on us. And that leads us to something rather intriguing. We know that epigenetic information is passed on from parent to child. It has to be. Azim's work shows us that. It must be passed on from parent to child. But we also know that epigenetic information can be affected by the environment, the gene, metabolizing the gene. If you take those two bits of information, both of which are undoubtedly true, you can make a really obvious hypothesis, which is, in that case, if we respond to the environment via epigenetics, and we know epigenetics is influenced by the environment, sure, could we pass on environmental responses to our offspring? And that seems like a really obvious, sensible hypothesis. But there's a problem. And the problem is this guy. Anyone know who the man with the really quite impressive sideburns is? Lamarck. This is Jean-Baptiste Lamarck. Now, he predates Darwin. And because biologists, we're very badly trained in history. And so we tend to hear the word Lamarck and go, Lamarck, Egypt. What a fool. Because Lamarck was trying to explain why new species developed. And his theory was, if you look at giraffes, the ancestors of giraffes were wandering around on the savannas. And some of the giraffes would stretch their necks a bit to get the juiciest, most succulent leaves. And just like a bodybuilder getting bigger muscles when she exercises at the gym more, the giraffes' necks would get longer because they were stretching them more. And then when they reproduced, they passed on that longer neck to their offspring. This is the inheritance of acquired characteristics. Now, the inheritance of acquired characteristics is not right. Okay, it is not the way that this creation of new species happens. It happens because, as Darwin worked out, some of the ancestors of the giraffes would naturally have slightly longer necks. They would be at an advantage because they could get to the more nutritious food stuff. They would pass on that advantage through a mechanism that Darwin didn't know about, of course, which is through passing on their genes. But, that Darwinian Mendelian model, that's the one we all work with for evolution. And I'm not for a moment suggesting it's wrong. It is absolutely right 99.999% of the time, but it's the 0.0001% that's the fun bit. Because recent research, and actually it's really well established now, shows unequivocally that just once in a while Lamarck was right. We pass on species can pass on acquired characteristics, and they do it not by changing their DNA sequence, they do it by passing on particular epigenetic modifications to their offspring. And this is my favorite example. Although if you're really fond of mice, possibly look away now. Okay? So researchers took mice, we'll call them F0, they're the starting generation, right? So they took these mice and they exposed them to the smell of cherry blossom. And every time they exposed them to the smell of cherry blossom, they gave them a mild electric shock. Now, after a while, the mice would smell the cherry blossom and they'd shake with fear. As to be fair, I think I would as well under similar circumstances. It's a classic conditioning experiment. The mice had learned to associate the smell of cherry blossom with something horrible about to happen. Then they allowed these mice to breed. And they let them breed, and they got the offspring, the F1s, and they exposed them to the smell of cherry blossom. No electric shock. They shook with fear. They knew that the smell of cherry blossom meant something awful was about to happen. And this happened in a really high percentage of the offspring, far too high to be caused by mutation. It is caused by epigenetic changes that were present in the parent and that developed in the offspring. The really clever thing about these experiments was that the researchers knew exactly which cells in the brain to look at and exactly which genes to look at and exactly which were the most likely type of epigenetic modifications to look at. And they were able to show that the ones in the offspring that shook with fear had exactly the same pattern of acquired epigenetic change at this particular gene associated with stress and fear in the same cells in the brain as their parents. It's extraordinary. Not suggesting for a moment the Darwinian model is wrong, it just gets tweaked a bit occasionally. You'd think, actually, if you go on Twitter or its more savoury equivalents these days, and you do something where you go like, occasionally the mark was right. It's awesome. You start these huge fights. Just brilliant. I used to do it for a laugh when I was bored. If I was at a particularly dull conference or a work meeting or something, I'd do it. It was great. So we now have a situation where we know that mammalian reproduction is completely dependent on epigenetics. We know that sometimes you can pass on epigenetics in a way that gives your offspring a bit of a leg up, because essentially being pre-programmed with the knowledge that something is dangerous is quite handy. And there's something else which epigenetics does, and that is it makes life for half of us in this room possible. Without epigenetics, women don't exist. Females, mammals don't exist. And if we don't, boys, neither do you. No offspring. This is because in mammals, in humans, actually all mammals, we're very genetically different, males and females. I mentioned it right at the beginning. All us ladies, we have those two big yellow chromosomes. We have two X chromosomes. As I mentioned, all the gentlemen, you have an X chromosome, good for you, and a Y chromosome, very small. Very small chromosome. Tiny, very few genes on it. I know, it's sad, it's very sad. The X chromosome, of which women have two, is huge. Thousands of genes on it. This creates a really, really big problem at the cellular level. Most of our chromosomes, so our chromosomes, the way our DNA is organized, come in 23 pairs, and 22 of them are called autosomes, 22 of those pairs. And they're exactly the same in men and women. They're exactly the same size. So if you choose a random gene on one of these autosomes, the levels of expression in a male cell will be exactly the same as the levels of expression in a female cell. But if you look at the X chromosomes, women have two. And so they will produce twice as much of a protein from any given gene on the X chromosome as a male. You can't have thousands of genes expressed twice as highly in cells and there not be consequences for that. It's just absolutely biologically catastrophic. So how do we get around it? Well, the way we get around it is that women switch off one of those chromosomes. So that essentially we end up with just one active X chromosome. And so we end up expressing the same number of genes as the men. Sorry, we end up expressing the genes at the same level as the men. And it's the most extraordinary process. It absolutely depends on those huge amounts of epigenetic change to the DNA itself, which you remember I told you compacts the DNA? What happens is one of the X chromosomes gets covered, absolutely covered, in that particular modification, and it condenses the X chromosome. It actually distorts it and it switches off all the genes on that chromosome. And it's an incredible thing because it happens when we are tiny embryos. So we're only about 128 cells big when this happens. In every single cell of that embryo, in the space of a few hours, one of the two X chromosomes in a female embryo gets switched off, completely randomly. One of the two gets switched off. And then the embryo keeps developing, and every single daughter cell inherits that same switching off as an X chromosome. Every single one. So you get 70 trillion cells And all the daughter cells that arose from, choose a random cell there, all of its daughter cells would have switched off that X chromosome. Choose another cell, that'll switch off the other X chromosome. This happens in the space of a few hours. You can actually see it in white blood cells. If you stain them with the right thing, then this bit here, that little thing there, is called the bar body. And it is the X chromosome that has been hypercondensed, and you can actually see it down the microscope. And of course, you only see it on white blood cells from women, not from men, because you don't switch off your X chromosome. And it lasts for the whole of our lives. So something that happens when we're 6,428 cells big, even when we're 70 trillion cells big and we're 80 years old, we've still got exactly that same pattern of having switched off one X chromosome in our cells. It's incredible. And we couldn't survive without it. It's an amazing phenomenon. And it also has other consequences. Has anybody here got a tortoiseshell cat? Or has ever had a tortoiseshell cat? You do, you do, you do, you do. Right, I'm guessing your cats are female. They're all female. That's because the tortoiseshell pattern is created by X inactivation. The colour, the genes for ginger and black fur are carried on the X chromosome. And they're just different versions of the same gene. So if a cat embryo, female cat embryo, has the ginger gene on one X chromosome and the black version of that same gene on the other X chromosome, when it's at that stage of being 6,428 cells big, half of those cells will randomly switch off the X chromosome that carries the ginger version, leaving only the black version active. The other half, on average, will switch off a copy of the X chromosome that has the black version of the gene. So only the ginger version can be expressed. And as the embryo develops, those cells pass on the same pattern to their daughter cells, so you get patches of black where the ginger X was switched off and those skin cells were created. You get patches of ginger where the ginger cells were the black one. You know what I'm saying? Yeah, half and half, yeah. So almost all tortoiseshell cats are female. Occasionally someone will say, mine's a male, and I'll say, it's infertile. And then they go, oh, yeah, it is actually. It's different, it's the same phenomenon, but has a slightly different basis. So I love that epigenetics, we can use it to explain absolute fundamentals of life, and we can also use it to explain things as daft as the colour of cats. Incidentally, if you have a tortoiseshell cat, do not try and get it cloned, because it will not come out looking the same. Because when you go through the whole when you go through the whole cloning process, both X chromosomes get switched back on, and then they both get switched off again, but in a totally random pattern. So you end up with a cat that looks nothing like the original one, which I love. Some idiot spent $30,000 cloning their cat. Look nothing like the original. They might as well have gone to the local cattery. They really might. Epigenetics is a really fun, really entertaining field, but one that we have had enormous problems as scientists engaging with. And the problem is not really a technical one. The problem is a cultural one. You still see ridiculous fights about epigenetics going on. I mean, there's a huge thing in the New York Times a few years ago about, is epigenetics important? And people go, no, it's not important, genetics is important. And other people say, no, it's not genetics, it's transcription factors, which are these special proteins and so on. And it was ludicrous. The reason is, I think, because biologists, you can get through your entire biological education, you can get your PhD, you can become a professor, and at no point will you have been forced to study the history or the philosophy of science. And so you can come out being technically brilliant, but never actually having thought about how your discipline works. We have a really terrible tendency always to assume the first thing that we find is the most important, and everything else is irrelevant. We were talking over dinner about exosomes. We've known exosomes exist for ages and ages, but we didn't know what they did. So we just went, well, they can't be important. Of course they're important, but we didn't know what they were. So we didn't know why they were important. There's a different type of histone called H1, right? Which, you know, I showed you that DNA goes around the 8 marshmallows, then it carries on and it goes around the next 8 marshmallows. Well, actually, what there is in the middle, where it just kind of goes like that, is another marshmallow, a slightly different color marshmallow called histone H1. And for years, people went, no, it's not important. There are millions of copies of that protein in the cell. What are the chances it's not important? And now we're starting to realize it is, just we didn't know. So we fall into this fallacy of, we think the first thing we find is the most important, and then we get very defensive about it. So we go, no, it's all DNA. We know it's all DNA because DNA must be the most important thing because we found it first. It's a ridiculous way of thinking. We also put stuff in boxes. So you'll literally have people going, genetics is the most important. Somebody else going, transcription factors is the most important. Somebody else going, epigenetics is the most important. They're all really important. If one of those bits goes wrong, everything goes wrong. We have to stop thinking of stuff in boxes. And we have to stop thinking of stuff in pathways. We are trained to think A goes to B, goes to C, goes to D. And we are trained to think in simple systems because we can recreate those experimentally. What we forget to do is to remember that we do that to understand a little part of the system, but that doesn't mean that's what's actually happening in cells and bodies. Cells and bodies are much more complicated, and it's all cycles and they're all interacting with each other. We forget. And so it's incredibly important that we train the next generation of biologists to be much more thoughtful about the systems that they're really interrogating. So I hope I have convinced you that epigenetics is fun and interesting and weird. There is a lot more I could have told you about. Epigenetics is absolutely crucial in plants. For example, winter barley has to be planted in winter because if you plant it in the spring, it will never set flowers and seed heads. And that's because the genes have to be exposed to cold. And we actually understand really, really well why that is. And it's all epigenetics. It's all about particular epigenetic modifications get put on in response to cold. Epigenetics plays a role in aging. It's not the only thing that's important in aging, but it definitely plays a role in that. And it also plays a role in something else. Would you like to guess the name of the sheep? Dolly the sheep. The world's first cloned mammal. I always have to point out at this point, Dolly is dead in this picture. People sometimes get the impression that Dolly was just so up herself and so full of herself that she refused to walk anywhere and just, I have my car. Dolly is dead and being taken to her case in the National Museum of Scotland. First cloned mammal. To be able to clone something where you take the nucleus from an adult cell and you revert it all the way back to being an embryo cell, you have to remove, I say you, the system has to remove huge amounts of the epigenetic modifications that have been established all the way through life, and then put on another batch of epigenetic modifications so that it can start developing as an embryo again. When you're doing something like that, the system that our cells use is pretty good, but it's not designed, it hasn't evolved to reprogram adult nuclei. And so it can't get rid of all the adult modifications, and it can't always put on all the right embryonic epigenetic modifications. This is why cloning is still very low success, and why often the cloned animals are less healthy than the original animal. It's because the epigenetics hasn't quite got sorted out. So there is so much more I could tell you, but we don't have enough time. But what we do have is a very nice person outside. Why is this stopped? Don't stop me. We have a very nice person outside selling some of my books. So you can find out more about it from out there if you'd like to. But for now, that's all I'm going to say.
I'm in the world of cosmetic science, and I'm very aware that in Cambridge recently, they took an old skin cell and they took it back to being a young skin cell. This really is exciting science.
It is really exciting, and the older I get, the more exciting I find it. Because there's only so much that one of you they can do. It is really exciting science. One of the things that you can do is in cell systems is you can reintroduce certain genes and overexpress them at a very high level. And it's extraordinary. Just 4 genes can push a cell from being adult right back to being embryonic. It's extraordinary. It was the guy who did it got the Nobel Prize. It absolutely, understandably, it was an extraordinary feat. We're trying to find ways of doing it chemically as well, because that's a lot in here. The thing I would caution, though, is that embryonic skin cells, great. The only problem with cells that express lots of the genes that we associate with embryos, those genes that you associate with embryos, when they get reactivated when you're an adult, are very typically associated with cancer. We have to be very, very careful about reprogramming the epigenetic state of cells, because if we go too far, there is a risk that what we will get is cancer. Doesn't mean it will always happen, but I agree, it was extremely exciting research. I also, unfortunately, occasionally see research that goes, we added this compound, we added something to these cells and their epigenetics changed, therefore we now have the fountain of youth. And it's like, no, you really don't, because epigenetics changes all the time. I was once asked to review one of these papers, and it's the only time that an editor has come back to me saying, I've never been asked to review before. Because I was sent this one that was actually in your field, but by a company nothing like as good as yours. And it said, we added this compound, we had epigenetic changes, we think this is going to drive youthful skin. And I had to respond saying, actually, they could have sprinkled cheese and onion crisps over it. They probably still would have found epigenetic changes, because our cells change all the time epigenetically. So we have to be, the bit that's really difficult is working out, just because the epigenetics has changed, has the behavior of the cell changed? Because there's massive dynamism in the system. But I agree, the Cambridge work was very exciting.
I mean, talking to the younger people in the audience, there have never been a more exciting time to be in science than the time we're in now. We are just...
It is a really fun time, because we also have a lot more tools, and we have the ability to handle huge data sets that we couldn't do before. Unfortunately, it means that all the mathematicians are wading in, but we kind of have to invite them to play nicely. People who are skilled at both biology and maths are going to be the ones who are massively employable and make the huge difference to research and to making the world a better place. I love curiosity-driven research. I think it's beautiful. I think it's a wonderful cultural activity. I think we should support it massively. But in my own career, the thing that I've found really fun is taking curiosity-driven research and turning it into things that improve the world. And there are more opportunities to do that than there's ever been. But don't get me started on that. That's a whole different
Well,
talk.
there's two words, AI and omics.
AI and omics. AI omics.
Two big things. So very exciting. There must be more than me. There must be questions out there you've got. So
Yeah, those people who have an extra chromosome,
Yes.
are those genes evenly distributed, and what gets switched on and what gets switched off?
Right. By the people who have an extra chromosome, oh, I need to be over there, or the mic can't hear me. Do you mean people who have an extra sex chromosome, like an extra Y, or do you mean people with Down syndrome, for example?
Well, I was thinking sex.
Okay, so you get people where you get women who have like XXX, right? You get men who have XXY. Now, if there's a Y chromosome, you will always be phenotypically male, okay? What's interesting is that if a woman has 3 X chromosomes rather than 2, she will turn off 2 of them. So she's only ever left with 1 active one. If a male has 2 X chromosomes and a Y, his cells will turn off 1 of the chromosomes. The system has evolved so that no matter how many X chromosomes are in a cell, only 1 will be active. Doesn't mean those people are completely asymptomatic. There's a small region at the end of an X chromosome that escapes inactivation. And that region of the X chromosome tends to be associated with height. So men who are say XXY, they tend to be very tall because they don't manage to switch off that bit of the chromosome. And actually women who have multiple X's tend to be very high. You also get women who only have 1 X chromosome in their cell. They don't turn it off. But what happens is they tend to be very short because they've only got 1 set of the tallness genes. So if you have an extra copy of chromosome 21, for example, you have Down syndrome. And even though chromosome 21 is tiny, Down syndrome is a very dramatic phenotype. If you have an extra copy of an X chromosome, your phenotypic change is actually very small, considering how huge the chromosome is. What you do, however, tend to find is that people are infertile, and it's because people with extra or insufficient X chromosomes are infertile, and it's because when the cells are dividing to create eggs or to create sperm, they can't separate and segregate out the chromosomes properly. But no, the X chromosome is huge, so the autosomes tend to be numbered in terms of their size. If you have 3 copies of chromosome 1, you can't survive. You don't even get anywhere near the term as an embryo. It's only in the X chromosomes that you can do it. Does that answer the question? I think you had a question.
Yes. We used to have chinchillas that
Okay.
were from the rescue centre.
Yeah.
When they arrived, they couldn't jump around or walk around, or they couldn't bounce on anything. Once they'd been in our kitchen for a couple of months and bounced, they would behave just as though they had been out in Patagonia bouncing around on rocks. Yet they had never actually experienced that.
Yeah.
Where did that come from?
Okay. I'm going to make stuff up now. And also, I should warn you that every time I make stuff up, I will make it up in different ways, but the answer always comes out as it's awfully complicated and there's loads of stuff we don't know. So here goes. There's lots of innate behavior which can be suppressed. Now, it can be suppressed by things like epigenetics, but we don't know. It can also just be suppressed by if things have been in animals or humans raised in very high stress environments. Our expression of the stress hormones that make us very stressed go completely wrong. So it could be that those are getting reset. Animals will naturally tend to revert to natural behavior. Humans, we're very different. I mean, one of my favorite quotations, and unfortunately I can't remember who said it, is that the human brain is the most complex 1.5 kilos of matter in the known universe, because it's billions of cells forming trillions of connections. And then you also have all the epigenetic modifications to genes and all that sort of thing. What we do know is that in humans, and actually we have pretty good data from rodents as well, that it's actually the very early life experiences that are most damaging. We tend to bounce back better emotionally from later life experiences. So certainly in terms of trauma, we bounce back better. So in other words, I have no clue, but I really like the idea of the chinchillas bouncing around your kitchen like they're in Patagonia.
It was a wonderful science. It really was.
I bet. I know, I keep chickens, we get rescue chickens. And they arrive and they're miserable, etc. And about 3 days later, they will be destroying your lawn quite happily like normal chickens. It's incredible.
These genetic markers, what are they actually made of? Are they made of protein?
No, they're made of very small chemical groups. So the ones on DNA are almost 99% of them are methyl groups. They're just a carbon and 3 hydrogens.
Oh, right.
Yeah. And they're all the ones on histones are more variable, but they tend to be really simple molecules like acetyls, methyls, phosphates, etc. They're tiny. What's important is that there are proteins that can recognize them. So there are proteins that can tell the difference between a histone that has a methyl group on it and a histone that doesn't. And those are the proteins that start the cascade of what happens in response to having those epigenetic modifications, because otherwise they'd just be decoration. But actually it's the fact there are proteins that can recognize them. So there are proteins that put those on, the epigenetic modifications, there are proteins that take them off, and there are proteins that can actually recognize them and bind to. So they're referred to as writers, erasers, and readers. What makes the epigenetic system really, really annoyingly complicated is that one protein may have multiple functions. So it may have a reader domain that can bind to a particular modification, like a methyl group in a particular position on a histone, but it may also have another domain in the same protein that is able to, say, take off a phosphate group. So it can change. I know, it's weird, isn't it?
When I learned about the basic molecular biology was in its infancy, and they talked about nonsense codon. Miles and miles of nonsense.
Right, yes.
DNA.
Not nonsense.
So I guess it probably wouldn't be.
No.
So many.
Exactly. So this is another area that's been hugely controversial. In fact, of my 3 books, the one that got me the biggest kicking was the middle one, which was junk DNA, with various people going, if I'd been writing this book, I wouldn't have written it like that. And it's like, well, that's fine, but you didn't write the book, so sure, go on. Yeah, you can tell I take criticism really well. there are massive stretches of our DNA. Only 2% of our DNA codes for proteins. And for the longest time, despite what the critics said, the consensus was the rest of it was literally just junk. It did nothing. We now know this is completely not correct. There probably are some regions that don't do much. And we have millions of copies of old viruses in our genes and in our DNA. But actually, we now know that lots of those areas, even though they don't code for protein, they control how protein is coded for. So they don't code for it themselves, they control things. One of those junk regions is actually the first step in switching off X chromosomes, which we were talking about. But they do loads of other things. We're now recognizing that some of them are really important in driving cancers, for example. And all those viral ones, the ancient viruses that are embedded in our genomes, if you mess up certain bits of the epigenetic system, they all come back to life. So the epigenetic system and the junk system work really closely together. They're highly evolved systems. It's a very weird thing, our DNA.
So, do we have any more questions? Yeah.
So how does cellular differentiation occur in the developing embryo?
How does cellular differentiation occur? Is that the select? It seems really weird, doesn't it? Because you get the cell that's formed when the eggs and the sperm fuse. I always find it really funny that I can tell anyone under the age of 40 is going, my parents had sex, don't horrible, I don't think about it. The egg and sperm fuse, and you've got one cell there, and really within a few rounds of cell division, it's already starting to change. And that seems really bizarre because you only had one set of genetic information. And our understanding of how all of this happens is still quite poor. But what seems to be the case, and the best understanding we have at the moment is, so that one cell, you've got the nucleus, you've got the cytoplasm, all the proteins and other stuff around it. And that cell divides. And for decades, we focused only on what was happening in the nucleus. And we assumed that what was happening in the cytoplasm was just a sort of bag of liquidy stuff that wasn't important. Now it's quite clear that what's happening is as the embryo is starting to divide, so one cell becomes 2, becomes 4, becomes 6, just because you can never have perfect separation in the cytoplasm, you will end up with a situation where one of the cells has slightly more of a particular pre-existing protein in it than one of the other daughter cells. That starts influencing the gene expression on the nucleus in that cell, and that starts influencing the epigenetics. And the more the epigenetic system gets activated, it responds to these changes. And so you start getting this divergence. But it almost certainly comes from this initial asymmetry in which half of a dividing cell the proteins end up in. And for the longest time, we ignored the cytoplasm. And the reason we ignored it was we didn't have techniques that are sensitive enough to investigate it. So that's the other thing that happens in biology. We are very driven by the questions we can address. So we focus on the things we can technically look into. Much easier to examine DNA than it is to examine proteins for all sorts of reasons. So we think that's how the cascade starts, and then things just start drifting further and further apart. Bit of a halfway question. Go on, you look cynical. Why are you looking cynical?
It seems to me that for example, the liver, you have a cell that turns into a liver cell. It's the adjacent cells that also turn into a liver cell. What determines how the cells develop is their geographical relation to other cells.
Yes. Yeah, that is an awful lot of it. The difficulty is that in terms of very precise embryology, we are still at a very descriptive phase. And it's because our molecular techniques are not sensitive enough yet to allow us to probe every step of the mechanism. But some very exciting work at the moment is managing to do things like take mouse embryos, and they're managing to map exactly how the brain is developing. You know, which cells give rise to which other cells. And a lot of it will be based on the geographical relationship to other dividing cells in that system. And we do know certainly from things like insects that sometimes just one gene, you can get the most amazing phenotypes if that gene is mutated. You can get fruit flies that have legs coming out of their heads, etc. So these are really, really subtle systems, but it does seem that a lot of it is driven by asymmetry initially. So once cells start to get different, they get more and more and more different until they get to a point where they stop becoming so different? And I would argue that we actually don't understand that stage very well. Why do they not just keep on going more and more and more different? Why do they, they get to be liver cells and they stop? They don't just keep becoming mad, weird cells. And actually, we don't understand that process very well at all. Give it 10 to 15 years and we will. And by then, the cosmetics will be much better as well. So I'll be here again to give this talk, but I'll look at least 25 years younger.
They do say, don't they, the first person who's going to live to 1,000 has already been born.
They do, and I pity them.
They do
Yeah.
have good skin. I think.
And they will have good skin.
Hope in a bottle, as they say.
Indeed.
Any more questions?
Is it completely random which of those X chromosomes is turned off?
Totally. Absolutely, totally. You've got 2 X chromosomes, and there is this gene called exist, XIST. It doesn't code for a protein. So the DNA, so the usual sequence is you have DNA, your genes, they make simpler copies of themselves called RNA, and then RNA codes for proteins. Exist is a gene that codes for an RNA, but the RNA never codes for a protein. And what seems to happen is when you've got the X chromosomes at this particular moment, they start to express exist. We're not quite sure what tells them to express exist. And the one that expresses slightly more exist than the other starts binding the exist to itself, and that starts a chain reaction. All the exist goes to that chromosome, spreads along. So the exist RNA spreads out along the X chromosome, and then that attracts the epigenetic modifications. But it's totally random, entirely random. There is no driving force behind it whatsoever.
I have a question, very naive question about the tortoise shell.
Oh, they're always the difficult ones.
Oh, sorry.
Please don't ask me why some of it's white.
It's 50-50 chance going on.
Mhm.
Yet the cat's whole ear was ginger in one side, and
So,
can that be explained?
yeah, so it's 50-50, right? So it's 50-50 in the early embryo, right? So we'll just, so 50% of the cells end up, let's call them ginger, and 50% end up
Yeah.
black. I know that's a terrible way of describing it. They will have, every time a ginger cell divides, it will create another ginger cell, right? And same with black ones. It will just so happen that based on where they are, on some cats, the ginger skin cells will be the ginger skin cells that happen to go over the ear. In other cats, it might be a black one. And in other ones, it will be that part of the ear is ginger and part isn't. It's entirely random depending on where they are in the embryo when they switch off one of the X chromosomes. So it's a very, very random process. So that's why no 2 tortoiseshell cats will look the same, because it's
Is
spectacularly
it
random.
that humans prefer cats to have a sort of largely ginger ear or a
No,
largely...
we can't really control it. We can't control, once you've got a female cat who has black on one X chromosome and ginger on the other, that cat will turn out in a way that we simply can't predict. Be quite a good way of running some sort of lottery, but we can't predict it.
I still haven't understood why they aren't just completely mottled.
Some are. So some tortoise shells you'll see, and you'll see that they're the black hair next to a ginger hair next to a black hair,
Yeah.
but it's because coloration, the cells that give rise to the skin, they don't give rise in terms of there's one cell and it creates all the skin. There are multiple cells that create different patches of skin. So there's a condition where it's very difficult to sweat. It's pretty much lethal in men. It's an X-linked condition. Women can have the same condition, but because they will end up with a situation where one of their X chromosomes codes for the healthy gene involved in sweating and the other one is broken. When you look at what happens in those women on their skin, they have patches that can sweat and patches that can't. And it's exactly this thing, different embryonic cells give rise to different bits of the skin, but they will all give rise to clusters of it. It's easier to draw, honestly. I apologize, I'm not explaining it very well, but that's why. It's because cells will give rise to clusters of skin, not just to one there, one there, one there, one there. So that's why it happens. You very occasionally find that this has enormous consequences in humans. So there's a very, if you're a genetics nerd, there's a very famous case, which is female identical twins. And one of them had the disease Duchenne muscular dystrophy. It's phenomenally uncommon for women to have this. So Duchenne muscular dystrophy is caused by a mutation in a gene on the X chromosome. Now, if a boy inherits a mutant copy of the gene, he has one mutant copy and then he doesn't have a normal copy because this is not carried on the Y chromosome. So all the muscle cells in his body go wrong. And so the boy ends up with muscular dystrophy. It's a really devastating condition, really awful. Women never really show the symptoms of muscular dystrophy because they have two X chromosomes. Yes, one can have the mutation and one may not have, the other one doesn't. And so when their muscles are being generated, typically what happens is half the cells in their muscle have the normal version of the gene switched on, half have the mutant version of the gene. But because muscle is a tissue that forms fusions, they end up with enough of the normal protein being expressed in their muscles that they're fine. What happened with this pair of identical female twins is that one of them was perfectly healthy and the other one had severe muscular dystrophy. And the assumption at first was that there must have been a mutation in the one who was affected. It must have happened very early in her embryological life, but actually that wasn't what happened. What happened, she was just really unlucky when her X chromosomes were being switched off randomly at that 64, 128 cell. All the ones that gave rise to muscle, all of those cells, just by sheer fluke, switched off the healthy version of the X chromosome. Absolutely random process. And so she ended up severely affected and her sister was absolutely fine. So it's an entirely random process.
Okay, any more questions? I'm guessing that if our delegates here would like to buy some of your books and read them slowly, and I think you wouldn't mind signing.
I wouldn't mind signing them at
Then
all.
they could perhaps understand some of the complications. It is very
They are page turners though, they're page turners, honestly.